All right, good afternoon. Let's go ahead and get started. So, not really any new announcements. As you know, you have exams coming up. So, this Thursday at 5 o'clock, this pre-midterm will become available on Canvas. Make sure you have scheduled yourself to take this. Once you start taking it, you can't stop it. Those 80 minutes will start clicking, and that is your opportunity to take the exam. So make sure you have a distraction-free period of time to take this exam. Don't wait to the last minute. If you wait too long, you may not have 80 minutes left. If you start taking at 4 o'clock on Saturday, it's not going to go the whole 80 minutes. It's going to shut off at 5 o'clock. So take it. Remember, you only need to get half right to get full credit for this, okay? So don't put it off. To make sure that you will be able to see the figures that are going to show up on this exam, I have put this practice online exam. 120 of you still have not looked at this practice online exam, so you like to live on the edge and hope that your browser's going to work. Once this exam opens, I'm not going to be able to help you with technical difficulties. If anyone has a technical difficulty, figure it out now, and you can definitely get a hold of me before the exam starts, and we will figure it out. Make sure that you are able to see the questions in your browser. The IAs will then be holding a review session in this classroom on Sunday. We have set it to podcast, so hopefully that will also be available for you if you can't make it here personally. That review session is entirely to go through all the questions that were on the pre-midterm. The pre-midterm is exactly reflective of what you're going to see on the actual midterm. So get used to these types of questions. The material that's going to be covered, and you will be all set for the midterm, which will be next Tuesday at this time in this class. Any questions? Yes? Do you think it will actually take us? I think it will vary by person. I can say last year, the quickest people were done in 15 minutes. The longest people took 80 minutes. So it's 20 questions. Pace yourself. You should have time to go back to ones you had questions about. But yeah. All right, a lot of chatter today. Okay, you guys are excited, right? All right, so let me give you a little. Uh-oh. What's happening? Oh no. Does this mean the internet went out? All right, try again. That is so weird. All right. You're seeing what we're going to go through today in reverse. All right. Let's see. All right. So this is what you're going to see when you open up this pre-midterm quiz. It is going to have these instructions. It's going to cover everything to the end of class on Thursday, so lectures 1 through 9. There's going to be 20 multiple answer choice questions. Each one's worth 2 points, so 40 points total. You have 80 minutes to do it. For their multiple answer, multiple choice. So A, B, C, D, all of them could be correct. At least one is going to be correct. Choose all and only the ones that are correct. For this pre-midterm only, not for the midterm next Tuesday, but for the pre-midterm, you can use any materials that are associated with this 2025 BIM 100 class that you are taking right now. So you can refer to your notes. You can look at the lecture slides, homework assignments, the textbook. I highly recommend that you don't count on that for every single question, or you will definitely run out of time. The questions are more application of what you have learned, so you're not going to look up something and find an exact answer in there. But if you do need to refresh your memory, like, oh, what does that term mean? Go ahead, look it up. This is actually a study guide sort of way to help you prepare for the actual midterm. Most definitely, you cannot communicate with another person, an AI agent, have someone else take the exam for you. I love Canvas quizzes because they keep a very nice digital fingerprint of everything you guys do during the quiz. Don't do anything suspicious that causes a red flag. It will be investigated. You signed an integrity pledge. Do your own work. You only need to get 20 out of the 40 points to get 100% for this assignment. And then, like I said, the answers will be discussed in the review session. Does anyone have any questions about what to expect for this pre-midterm online Canvas quiz? Yes?
Would you be able to go back to it and use it as a time, or will it be like answers off the show?
After it is over, at some point before the review section, I will post, you'll be able to look at your answers and see what you missed, so you know when you go to the review session. And then I'll also post a PDF version of it for you to bring to the review session to help you go through it. Question?
Are you going to post any other practice questions? Practice problems?
There will still be a homework 4 coming up on Thursday as usual, and so you'll have the opportunity to practice what we're learning tonight and on Thursday. Yes? It will be podcast. It is scheduled for podcasting. Anything else? Yes? This Thursday will be the last class of material. So lectures 1 through 9. If you look at the syllabus, you'll see they're numbered 1 through 9. Anything else? All right. This is what you will see next Tuesday when we hand out the midterm that you're going to take here in class. So it will be a written exam. You'll need to write your name on all the pages. For the actual exam, going through again, they're going to be multiple choice, multiple answer. You need to use permanent ink on that exam. So blue or black is fine. Once you have completed that, you're going to raise your hand, and then we will give you a bubble sheet, and you will transfer your answers onto that bubble sheet. They better match on the exam that you actually are handing in as well. You will hand in both of those things. For the bubble sheet, you can use pencil if you want. I know sometimes you make a mistake transferring. If you make a whole bunch of mistakes, we'll give you a new one. Don't stress about that. Very important, you need to bring a picture ID to class next week. It can be your UCSD ID, driver's license, passport, anything that has a picture of you and your name on it that will match the name on the exam that you are turning in as your own. Any questions about this? All right. Question for you. Which statement best describes your preparation for the exam? All right, pretty straightforward. 10 more seconds. Click in your answer. You'll get extra credit just for answering this question, right? It's a good deal. All right, put your final clicks in. Let's see how we're preparing. All right, good. So we have a lot in sort of this A and B realm. We have some jokesters here. How can you not know there's an exam? Hopefully this means you do now. A few of you have a bit of catching up to do, but you still have time. Even if you are in this D where you're just in class and haven't really done much yet. Oops, sorry. You still have time, right? Those of you who have been coming to classes, going to discussion sections, paying attention, doing the homework, you're doing everything you need to prepare for this exam. There's going to be no surprises. Only what we have talked about in class and repeated with clicker questions, with homework questions, is the type of material that you're going to be expected to answer on the exam. So don't despair. You still have time, but you really got to get your act together and start revisiting anything that you have missed. Any questions? All right, so let's review what we talked about in class last week. So last week we started talking about this second basic molecular mechanism, and that's the process of transcription. Transcription is using a DNA template to synthesize RNA. RNA polymerase is the main enzyme that does this synthesis. For the polymerase to know where to start transcribing a gene, there's got to be sequences, sequence elements, specific sequences that are going to be recognized by specific proteins to help position RNA polymerase II so that it can transcribe the gene. We talked about that there needs to, at the very least, be a core promoter element, the very basic element sequence that is going to be needed for RNA polymerase II and all of its general transcription factors, which we'll talk about today, to sit down near that +1 site and be able to start transcription. We talked about 3 general types of core promoter elements. For a gene to be transcribed, it needs to have one of those core promoter elements. So the example shown up here, highlighted in yellow, this is a TATA box. That's one of these core promoter elements. And this is really the most specific sequence to recognize. As the name suggests, it is TATA. It is a TATA-rich sequence that is always going to be located about between -25 and -35, counting upstream from the +1 transcription site. So the TATA box can be recognized because there's a specific sequence and it's at a specific position relative to the +1 site. What's the name of another core promoter element that we talked about? Yes? The CPG islands, right. And that P just stands for the phosphodiester bond. So you're going to see CG, 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 CG. And that's usually going to be located sort of a little bit distant from the +1, usually about -100, 200, -300. So it's going to be a bit further upstream. And you're just going to see this big string of C's and G's. And we said genes that have a CPG island, they're a little bit strange in that sometimes they have difficulty positioning polymerase in the right orientation. And so to some degree, you're going to get transcripts going in the opposite orientation from where you want it to go. We call that divergent transcription, going in opposite directions. And those transcripts that are going in the wrong direction, they usually are lowly abundant, they get degraded. The other thing about a CPG island is often there's not just like one +1 position, there's a little bit of heterogeneity where it actually starts. Usually doesn't matter for the protein coding capacity of the messenger RNA because you'll just have a slightly different 5' UTR sequence. All right, so our gene could have a TATA box, it could have a CPG island, and then what is the third type? Yes? Initiator, yes. And so this, as the name suggests, has a specific, not so specific, but has a motif at the initiation site. There will be an A at the +1, and then some pyrimidines upstream and a few other nucleotides that are pyrimidines or specific AGs or CTs into the gene here. So the initiator is sort of the least specificity. So if you're looking at a promoter sequence and trying to figure out which type of core is it, first look, do you see in the -25 to 35 region, TATA? If yes, then it's a TATA box. If you don't see that, look around -100 to -300. You see a whole bunch of C's and G's, then it's CPG island. If you don't see that, then hopefully there is an A at the +1, and we will call it an initiator. Question? Yes, yes, and because some genes have alternative transcriptional start sites also, and so it can have different core elements regulating different ones. So You need to have one. Sometimes there's more than one. Other questions? Yes, Lydia. Yes, yeah. So the ones that are going in the right direction, there are sequences in the messenger RNA that help the polymerase complex know that it's going in the right direction. All right, so in addition to having a core promoter element, there can also be a lot of other regulatory sequences or elements that can determine how highly expressed or potentially if the gene will be repressed. And if these are sequences that are closer to the +1 site, we call them promoter proximal elements. And if they are further away and usually longer, they are called enhancer sequences. We then talked about methods we could use to identify these types of regulatory sequences or elements, and we said we could build a reporter gene. And so a reporter gene simply is you have a coding sequence that will make a protein that you can detect. Green fluorescent protein. You shine UV light on your cells. If you see GFP, that reporter was expressed. And then in front of that protein coding sequence, you will put your promoter sequence of interest and ask, is it going to drive expression in my particular cell type or a particular tissue that you put it into? The next thing you might want to identify is, well, where are the sequences that are important for driving this expression? And the first thing we talked about is we can have this long promoter sequence, predicted promoter sequence, like let's say 2,000 nucleotides long. And we can just start doing 5' deletion analysis. So start chopping it shorter and shorter and shorter and shorter. And then just look, when do we see expression change compared to the full length? That might tell us, oh, okay, we don't need 2,000 nucleotides to analyze. At 1,000, we still get full expression. Now we want to identify the specific sequences that might be regulating expression. And for that, we use mutations. So you systematically start to mutate sequences in that promoter region hooked up to your promoter and look and see, does expression go up? Does it go down compared to the unmutagenized? If you see a change in expression, then you've hit a regulatory element. Any questions about that? All right, let's do a clicker question, see if we can interpret these types of experiments. So based on the results of the mutational analysis, so these black boxes is where we're having mutations, of this promoter gene, gene X, which of the following can be concluded? And so high numbers means higher levels of expression, lower numbers mean lower expression. All right, 10 more seconds. All right, final answers. All right, so a lot of love for B, but maybe it's more than one of the above. Maybe D is also a correct answer here. So let's go through each one of these. Who wants to volunteer their answer and why? Yes?
I said B, and the reason why is because in the -60 region, it says not of number 2. The moment that you have like that kind of mutation, there seems to be a decrease from 5 to 2 in terms of reported gene expression. So I was led to believe that there is likely a positive regulatory element in the -60 region, since it decreases when there's a mutation.
Yeah, absolutely. So if we look at -60, so which of our mutants affect -60? So we have mutant number 2 and mutant number 3 is changing sequence in -60. And in both of those mutants, our expression level has gone from the unmutagenized 5 down to 2. If we have less expression, we have lost a positive regulatory sequence. And what we can see is this region, the 60 region, it doesn't overlap with our mutant 1 or mutant 4, which is important because those two mutants, those regions that were mutagenized, didn't change expression. So we can really narrow down the region that has that regulatory element in that it must be sequences that do not overlap with mutant 1 or mutant 4. So certainly, yes, there is a positive regulatory element in the -60 region. Does everyone understand why B is correct? Question?
Can you explain it again? Like, I don't understand. I thought it was A. It was like I'm saying there's expression in all of them.
All right, so let's go through each one. So A, so let's see why A is not right. So A says all of the mutations affect expression. If I say affect expression, what does that mean? It's different than what we see in the unmutagenized. If we affect something, we have changed it, right? So the unmutagenized, no black boxes, this is our control here, it's not a mutant. We have an expression level of 5. So if all of them affected expression, we would have to have a number different than 5 for all of them. Does that make sense? It would go up or it would go down, right? That would be affecting the expression. But here we see mutant 1, we can mutagenize all this sequence here, and we have no difference from the unmutagenized. So mutant 1 didn't affect expression. Which other mutant didn't affect expression? 4, exactly. So we look at 4, when we mutate this sequence, no different from the unmutagenized. So 2 of our mutants had no effect. Question?
For B, why isn't it a negative regulatory? That's what I thought it was. From 5 to 2, and we're not messing.
Right. So if expression goes down when you lost something, then it must have been acting positively when it was there. If expression goes up, then what kind of element have we lost? A negative regulatory element. You have to think about what it's normally doing. So it's normally acting positively because when we lost it, we have less expression. Does that make sense? If you have higher expression than the unmutagenized, then it must have been a negative regulatory element. If you have lower expression, like we do in mutants 2 and 3, then we must have lost a positive regulatory element. Is that clear? All right, so moving along here, C says if all sequence upstream of the black arrowhead was deleted, the expression level would be 5. All right, so here's our arrowhead. Which direction is upstream? Moving towards -100 or towards +1? Upstream. Going negative, right? Right? Okay, so if we delete, all of this sequence, so do a deletion analysis, for example, would our expression level be 5? I'm seeing a lot of people shake their head no. What would it be? 2, exactly, because we already decided that in B, there was a positive regulatory element in this region right here. So if we got rid of that positive regulatory element, because we only have sequence now from like here to here, what would the expression be? It would be 2. We've lost the positive regulatory element. Does that make sense? D says there is a negative regulatory element in the -30 region. Okay, so we go to -30. The only mutant that affects that is mutant 5. And, ooh, what do we have here? We have 0. Did we lose a negative regulatory element? Positive. In fact, what did we lose? A core promoter element, exactly. So it was not more than one of the above. B is the only correct answer. Does that make sense? So when you're doing this type of analysis, what you always want to do is you want to look at where has expression changed compared to the unmutagenized. And then you go and see, well, where are the sequences that must have been changed to give me this score? Why in mutant number 2 didn't I circle this region right here, this like -80 region? Exactly, because it overlapped with mutant 1, and look, mutant 1, no effect whatsoever. So I can already narrow down that there is no regulatory sequence within this region. Same with in this section right here. This region in mutant 3, this little bit here, overlaps with mutant 4, no effect. So this region does not contain a regulatory element. Does that make sense? All right. Okay, so now let's move on to some new material for tonight. So we're still going to be talking about transcription by RNA polymerase II. But you remember I always told you that POL2 itself is this complex of proteins. And not only is RNA polymerase II a complex of proteins, but there are a lot of general transcription factor complexes that need to help bring RNA polymerase II, position it at the +1, and help start unwinding the template. So we're going to talk about the sequential binding of these general transcription factors. So these letters, so TF stands for transcription factor. Roman numeral 2, what do you think that stands for? It works with RNA polymerase II. And then the letter just has to do with the order in which it was discovered, but not necessarily the order in which it will sequentially bind to the core promoter sequence. So the role of these general transcription factors, along with RNA polymerase II itself, is to help position the polymerase and to unwind the DNA. All right, so we're going to talk about this assembly of the general transcription. All right, well, it says complex there, and then the abbreviation is PIC for pre-initiation complex. All right, so we're going to start off. We have our promoter DNA. In this particular example, it is a promoter that has a core TATA box. The very first complex of proteins that is going to bind to this promoter sequence is going to be transcription factor 2D, TF2D. TF2D is a complex of multiple separate proteins that all come together to bind at the core promoter. One of these proteins is called TBP. What does TBP stand for? TATA binding protein. It directly is going to bind to this TATA sequence. So TBP is the TATA binding protein. It itself is going to touch, it's going to bind to this TATA sequence and help position this whole TF2D complex. The other proteins that are part of this complex are generally called TAFs, which stands for TBP-associated factors. They're going to be more important when you have a core promoter that doesn't have a TATA box. Then the TAFs are going to help with these CPG islands and with Initiator. So the first complex to come in is TF2D. Once TF2D binds the core promoter sequence, TF2A and then 2B are going to join. And this assembly now is able to bend the DNA. And this bending of the DNA is important, helps put it in a conformation for the next factors to come in and bind. The next factors are going to be RNA polymerase II itself, which is multiple proteins together as a complex. And along with RNA polymerase II is TF2F. They are going to join this complex now. So now we have polymerase there. We're almost ready for transcription to start. But we need a few more factors. The next one's going to be 2E and then 2H. And 2H actually brings in some enzymatic activities. It brings in a helicase activity. So one of the proteins that is part of TF2H has helicase activity. Why do you need helicase activity? To unwind it, right. So you can find that +1, the template, so that transcription can start. Once the pre-initiation complex is opened up, is unwound, this helicase activity, we then have an open pre-initiation complex so that transcription can initiate. TF2H also has kinase activity. What do kinases do? Phosphorylate. What do you think is going to get phosphorylated? There's one part of this complex we've talked about before. Do you remember what it's called? The CTD, yes, the carboxy terminal domain. It is a part of one of the proteins in RNA polymerase II. Okay, so we have our open complex. We have all the general transcription factors in RNA polymerase II there. So nucleotides can start coming in, base pairing to the template, synthesis can begin. The 2H activity is going to phosphorylate that CTD, and the complex can start moving, transcribing the RNA. Most of the general transcription factors then release Pol2, so it can continue to transcribe down the gene. To facilitate this elongation, there are other proteins that are called elongation factors that come in and help the polymerase be processive and continue transcribing. Now, in many types of eukaryotic cells, RNA polymerase II actually often pauses after transcribing only about up to 100 nucleotides. And this is through the activity of several factors. One of them is called negative elongation factor. It's a good name for it. What it does is it binds to the polymerase complex and it blocks the nucleotide triphosphates from entering the active site. If you can't get the substrates for transcription, you can no longer continue to transcribe. And so what this can result in is that polymerase, it's already transcribed some of it, but it's sort of paused at the gene, ready to rapidly take off if there is a signal. It can also be sort of a proofreading step, making sure like this is a gene we want to transcribe, this is the level we want to transcribe it. And then when either the signal comes in or those checks have been made, there are elongation factors such as P-TEFb that come in, they phosphorylate NELF, kick it off, and then they relieve this inhibition so that elongation can continue. Any questions about setting up that pre-initiation complex and then shifting into elongation mode? Lydia? Yeah, so this isn't true for all genes, but some genes in our eukaryotic cells, it's thought to maybe get the polymerase ready for a signal that says, now we need high levels of transcription. And so you've already recruited all the general transcription factors, you open the chromatin, and you can have very high levels in a very quick period of time. Is there another question? Yes?
So do the general transcription factors like kind of call themselves to what's going on, and then do they call on like specialized ones, or do they have special proteins or genes that are calling them separately?
So most eukaryotic genes, in mammalian cells in particular, require all of these general transcription factor complexes to come in the sequence that we just went through in order for the complex to assemble correctly at the +1 site so you can get transcription. And that's a good question to lead into the next thing, which is like, oh, how am I going to remember this sequential assembly of all these factors? And so there's a very simple mnemonic that I use to remember this, and it is DNA bending to facilitate efficient helicase. So what does that mean? It means TF2D, that's where the D comes from. And the next one to come in is going to be 2A. So 2D, which has TBP as one of its proteins, that comes in first. 2A comes in. 2B is next, and that actually complex bends the DNA. And that is 2, Pol 2, facilitate. 2F is the next one to come in. 2E is next, an efficient helicase. 2H has that helicase activity. So if you can remember this mnemonic, you'll always remember the order, the sequential order in which these factors come in. If you do not have TF2D, you knock it out of the cell. Are any of these going to associate with a promoter? No. What we mean by sequential binding is you need D to be first, then A can join, only then can B join, then 2 can join, then F, then E, and then lastly H. Does that make sense? Yes? What does a 2 stand for? Pol 2, right? RNA polymerase 2. So the Pol 2 complex, 2, Roman numeral 2, 2TO. So other questions? All right, so for the rest of this class, we're now going to talk about a variety of experiments we can do to identify specific DNA-protein interactions. The first two types of experiments we're going to talk about are considered to be in vitro or in a test tube, meaning we know exactly the ingredients that we are putting into that test tube to ask if a specific sequence of DNA we've designed interacts with a specific protein or complex of proteins or even like a nuclear extract. And the first one we'll talk about is called DNase I Footprinting. You'll see where that name comes from when we look at the experiment. And then the second one is Electrophoretic Mobility Shift Assay, or EMSA for short, or sometimes we just call it a gel shift assay. And again, when we look at the experiment, you'll see where this name comes from, how we're looking for a shift that is indicating a protein has bound a segment of DNA. When we identify sequences or elements that interact with a particular protein of interest, of course, the next question is going to be is that relevant? Does that actually regulate transcription? And so we want to test the function of the interaction. And so this is where we can go in and ask, does this element or in this protein actually regulate transcription? And then finally, we will talk about an experiment that's considered to be more in vivo experiment. It's where we're asking whether a protein or a modified version of a protein interacts with a specific sequence of DNA in a cell, in a tissue, under particular conditions. And that is called a Chromatin Immunoprecipitation experiment, or CHIP for short. And so for all of these experiments, what we're simply asking is, all right, there's all these regulatory elements, regulatory sequences of DNA. The only way that they can be regulatory is if a protein binds and activates or represses expression, right? So we need to identify what are the protein DNA interactions that are regulating expression. And so again, this is just another way to say that question is here we have identified maybe through our mutational analysis using a reporter, this segment, this sequence is regulatory, it's activating my reporter. So is it activating because this protein that's expressed in this cell type is binding to that sequence? And so the first type of experiment we'll talk about is a DNase I Footprinting Assay. All right, so this is, remember, an in vitro experiment. So we're going to have very specific reagents we're going to put into a tube and ask, can a protein bind to a specific sequence of DNA? So when the protein is binding this DNA in the cell, the DNA is double-stranded, right? And so you're going to have a segment of double-stranded DNA, and you're going to use radiolabel to put labels so you'll be able to detect the DNA just at one end of your double-stranded DNA. So let's say at this 5' end of this double-stranded segment of DNA. After we go through the experiment, you'll see why you only want to have label at one end at one position. you're going to have at least 2 different samples, so 2 different tubes. In one tube, you're only going to have your double-stranded DNA. In the other tube, you're testing, this is your test sample, does a specific protein actually bind to this sequence of DNA? And so in this tube, you're going to incubate the DNA with your protein sample. And then to both tubes, you're going to add a limiting amount of DNase I. What does DNase do? Chops double-stranded DNA, exactly. The reason we put a limiting amount in is we actually want, on average, to get only 1 cut per molecule of DNA. If we put a whole bunch of DNase in, we're going to get many, 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 many cuts, and then all we're going to be left with is 1 nucleotide with label on it, and that's the only thing we're going to see. Instead, we want, on average, 1 cut, and if there's no protein in our tube, the whole DNA sequence is available to randomly be cut at various positions along the entire length of it. But if our protein binds to a specific sequence in that DNA, it's going to be protected from the DNase. So wherever that protein was bound, you will never get a cut at that position. So you will be missing cut pieces of DNA where the protein was protecting it at that position. All right, so after you have done this DNase reaction, you then isolate the DNA, so get rid of the DNase, get rid of the protein, and then you run it on a denaturing gel, so you do electrophoresis. Now you can denature the DNA because you just want to look at the different sizes of bands that you got from that DNase reaction. And the only thing you're going to be able to see are the fragments that had the radio label on it, right? So they are going to run based on size. Bigger size bands are going to run closer to the top. Smaller size are going to be towards the bottom. I also run a lane that has an M above it. What does M stand for? Anyone know? M? Marker? The marker lane? Sometimes you call it the ladder. It is simply a lane where you have bought or used PCR to create your own sizes of DNA. And this is just a marker to tell you that, any band that runs close to this 10 is going to be about 10 base pairs long. Anything close to 100 is going to be 100 base pairs long. All right, so let's look at our sample. So first we had our tube where we didn't incubate protein. So on average, we've gotten a cut at every position of this double-stranded DNA. If a cut in one of your molecules of DNA was way far away from the radio label, then this size band is going to run towards the top. It's going to be long size. Any that got cut closer to where your label is, it's going to be down here, right? Does that make sense? So you see now why you only want to put radio label on one side. If you put it on both sides, then you would get from here both a long and a short band. You would not get any sort of pattern you can make sense of. All right, so this is to make sure your control worked. You got bands that basically go across the entire segment of DNA. But now let's see what happens in our tube where we have had the protein incubated. In that case, we know that we are never going to get cut where the protein was bound. If the protein is bound, it is protecting the DNA from this DNase cut. Every other position, we will get cuts, but you will then see a space where you never got a size band that would be where the protein was bound. Where would these pieces of DNA run on the gel? The top, exactly. In fact, you see this big, big, bright band up there? That's because, remember, we used a limiting amount of DNase. So a lot of our DNA just never got cut at all. And all the ones that were protected and could never have a cut in this position, anything uncut is going to run towards the top. When we then purify our DNA, we are never going to have sizes that would run at these positions, and that's then what we call the footprint. It is the space where the protein protected the DNA because it was specifically bound to this sequence. if the protein bound closer to the label, would my footprint be further towards the bottom or further towards the top? Towards the bottom, yeah. If we had our protein protecting here, then we would get a few small bands, but then we would get a space here, and then you get all the longer bands instead. Does everyone understand where this footprint comes from and how this experiment works? Question? So if you have some proteins will leave you a footprint that could be 30 base pairs long, a bigger protein or bigger complex or the way it binds the DNA. Other proteins might only protect 6 nucleotides, and so you'd see a much smaller footprint. So from this type of experiment, you get information about the size of the DNA that was protected, how many base pairs were protected, and also the position. Where was that protein bound? In that example we just did, the protein is closer to the label, then we know that it is bound closer to these positions. If I tell you this is like position minus 100, then it's binding closer to position minus 100 than to plus 1, for example. So the experiment would definitely be limited by how big the pieces of DNA are that will run into the gel. So if you start to get more than several hundred base pairs long, it's hard to run a nice gel and separate all those bands out. Is that what you mean? Okay, yeah. If you throw in an extract and it covers the entire thing, then what would you see in your experiment? Only stuff at the top. Nothing would have been cut. You would just have like a big blob of uncut DNA. Question?
So our cut that we made to our tag should be like the same length or equivalent to what we see on our testing by our footprint, right?
So every place where the protein wasn't bound, you will get a cut. So like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, up to 30, and then you will not get any cuts here, and then you'll get pieces of DNA that are going to be much longer, up to 100 nucleotides here.
So like you could technically say like you could match it to this on the gel and then this to this.
So yeah, the whole point of doing this is so that you can identify that you know your sequence of DNA that you put in, right? We have a test tube, we created the DNA, we bought it from a company, we know its sequence, we know where we put the radio label. And so based on where we see the space, we know where the protein bound, and therefore we know the sequence that the protein bound also, right? Maybe this will become clear in the next clicker question that we're going to do. Question?
How does the DNase like know where to cut? Is it all just random?
It's random, yep, yeah. So DNase I just will cut DNA anywhere. There's a little bit of bias. Some positions it can cut a little bit better than others. But that's why you do your control, to make sure you get a nice pattern. Because if our control also had a space here, then that would have been a part of the DNA that for some reason the DNase I wasn't cutting. So you're always going to compare the control with no protein to when you added the protein. What's the difference? All right, let's do a clicker question on this. So the results of a DNase I footprinting experiment using double-stranded DNA. All right, I've only shown you one strand, but it's double-stranded. You know what the sequence of the other strand would be. And we've radio labeled it at this minus 50 T position right here. And then we're looking at it in the presence of various proteins and various cellular extracts. So based on these results, the data support which of the following statements? I can, but I'm hesitant to do anything with the clicker because
it kind of breaks.
Yes. Yeah. Yeah. Yeah, so sorry the clicker thing is covering it, but I think you can see that lane 4 says nuclear extract plus protein X. 5 says cytoplasmic extract plus protein X. I would try to move it, but the clicker system's so touchy that I want to baby it. 10 more seconds. All right, final answers. All right, nice little bell-shaped curve almost. All right, so C, people are really like C, but everything got at least a couple of votes. So let's go through and make sure we know how to interpret these types of experiments. So who wants to share their answer and why? Sure.
I chose A because like protein X, like I can see it was binding to the nuclear extract plus protein X. So there's like a huge part of it was bound.
Okay.
To the DNA there.
Okay, I understand your reasoning, but does this type of experiment tell us protein X is a nuclear protein? No, it doesn't give us any information on where this protein came from. So what can this type of experiment tell us? If the protein binds, do I even get a footprint? If it does, where does that protein bind based on the region that I give you here? And the size of the footprint, so how much DNA was protected. Tells me whatever protein this is, did it interact with the DNA, where in the DNA, and what size. Doesn't tell you what the identity of the protein is or where it came from. Does that make sense now? All right, so A is not correct. B, a lot of people like this. Protein X binds at position -10 to -20. So that'd be in this region here. Is that correct? Yes. Right, exactly. You got to remember where our label is and where we see bands. So protein X, does protein X bind this piece of DNA? Yes. We can compare it to no protein. We most definitely see a footprint. So we definitely got binding. Where does it bind, though? -40 to -30. -40 to -30. How did we get there? Well, let's see here. So with protein X, we get bands that are 10 base pairs or smaller. So that means we got some that were cut there. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then all of a sudden we have a space. -50, -10 is going to put us to -40. We have a 10 base pair footprint, right? So there's a block of 10 nucleotides. So -40 to -30 must have been bound by the protein in order to give us this footprint. And then every other position, including -10, -20, everything here has a cut in it. Does that make sense? You have to think about where is my label and what size bands am I seeing? So it doesn't bind at position -10 to -20. It binds at position -40 to -30. Does that make sense? We'll visualize that in the next slide too if it's not quite clear. C says the cytoplasmic extract has an activity that prevents protein X from binding the DNA. A lot of people like this. Why do you like this one? Sure. Yep.
So I chose C because if it were to be wrong, if it didn't prevent it, then we would see a footprint in the far right column.
Yeah. So we saw that protein X alone, so the only thing we have in our test tube is our double-stranded DNA and protein X. We got a footprint, so that means protein X can bind to that DNA. The only additional thing we've added in this tube is cytoplasmic extract. We lost the footprint. Something happened that we no longer, what could have possibly happened? Another protein bound what? If it bound to the same space as protein X, what would that look like? It would look like the footprint, right? Could bind to protein X and prevent it from binding DNA. Then we would get none. It could degrade protein X. We have no protein X left. We also wouldn't get a footprint. So C is the correct answer. Let's go through the rest of these. Protein X can only bind the DNA in presence of the nuclear extract. So we already said protein X alone in our tube, we get a footprint. What has happened when we add the nuclear extract? We get a bigger footprint. How would we get that? So there could be more proteins in here that are also binding this sequence of DNA. There could be an activity that modified protein X so that it binds the DNA differently, that it covers a larger space here. We don't know because we don't have a nuclear extract alone here. But we definitely can say that protein X does not need the nuclear extract to bind because it alone bound. And then E said binding a protein X protects 20 nucleotides of DNA. How much does it protect? 10. And so here's where it's really important to have your marker lane. Just refer to that, and you can see 10 base pairs were protected by the footprint. And then here's just, again, to visualize this. So you can imagine if you draw protein X at this position, -30 to -40, and then you got your DNase cutting. Some molecules you would have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We see those bands of those sizes 1 to 10, and then we see a space, and then we see the longer pieces. Does that make sense? On the exam, you'll have this here. Draw it out for yourself. Make it clear in your head what this means. All right. Now let's talk about the gel shift, or the electrophoretic mobility shift assay, also called an EMSA. So this is also to ask the question, in a test tube, does a protein of interest bind to a specific double-stranded piece of DNA of interest? And so you need, again, to have your radio-labeled double-stranded DNA. Sometimes we call this a probe. So your double-stranded DNA, you put radio label on it, and then you incubate it with no protein because you want to see in your gel where the DNA alone will run. And then you can test a variety of other proteins. If the protein binds to that DNA, it's going to be a bigger complex, right? It's going to run more slowly through the gel. It's going to be shifted in the gel. That's where the name comes from. It's a gel shift. An important thing here is you need to perform your electrophoresis in a non-denaturing gel. So what do we mean by denaturing? Right, you break the double-stranded DNA so it's single-stranded. And in fact, when we did our DNase footprinting, we did a denaturing gel because we just wanted to separate those naked pieces of DNA based on size. But here, why are we using a non-denaturing gel? What would happen if we denatured our DNA? Protein would bind, right? So we would see nothing. So the protein is bound to the double-stranded DNA. You want that complex to stick together so it can shift in the gel. So which of these test proteins would you say bound this piece of DNA based on these results? B and C, right, exactly. And this is very common where you don't shift all of the probe. You're usually in large excess of the double-stranded DNA. But most certainly, if you see the bands running higher in the gel, migrating more slowly, the only way the DNA which is what you're seeing, can run more slowly in the gel is if it's bound to the protein. Does that make sense? Question? Exactly. Very good point. So an advantage of the DNase footprinting over the gel shift is with a footprinting, we see the position of binding. We know where the protein bound within that sequence of DNA we put in. Here, the protein would look the same in the shift if it bound down here, if it bound down here. It's just shifting the DNA. So it is a way of saying that the protein interacted with the DNA, but not necessarily the position. One thing this type of assay is good for is for measuring sequential binding, meaning do you need a protein there for another protein to join the complex? And in fact, that is the topic of this clicker question. So above are the results of an EMSA assay using TATA box core promoter DNA sequence as the probe, and different combinations of your pre-initiation complex factors. So based on all of the data and your knowledge of the sequential assembly of the pre-initiation complex, which of the following are possible? So NP means no protein, so this is where our free probe binds. And then it's saying which of these samples. And when I say, for example, sample 1 is TF2B, that means the only thing we put in that tube is the probe, the double-stranded DNA, and TF2B. Got it? Ten more seconds. All right, put in your final answers. Oh boy. All right. So we have a variety of opinions on the answer here, so it's a good thing that we're going to go through this. Anybody really brave and want to share their answer and thought process? Yes. Yes.
I think it's C because I don't think you can find TF2B or DNA or TF2B or TDP. Like we needed to have it in kind of a sequential assembly altogether.
Right, so your thinking is correct. Remember, we have our knowledge of the sequential assembly of these general transcription factors, meaning that you can only get the next factor to bind if the previous factor was there. So what is our very first factor that binds? D. Remember our DNA bending to facilitate efficient helicase? So if TF2D is not there, can any of the other ones bind? One of them could, right? So what's TBP? TBP is TATA binding protein, which is a part of TF2D. All right, so let's jump to, so sample 4. Could sample 4 be TBP? No, because TBP binds directly to the TATA box. Would we see a shift? Yes, if we put TATA box core promoter DNA and TBP in a tube, we would definitely see a gel shift. Sample 4, are we seeing a shift? No. All right, let's go to sample 1 here with the same idea of what we were talking about, sequential binding. So sample 1, do we see a shift here? Yeah, so something bound. Can TF2B bind with nothing else in the tube? No. Because who did we say needs to bind first? 2D. Sample 2 is TF2D. Do we see a shift? Yes. Can 2D, this complex, bind to the DNA with none of the other factors yet? Yes. So B is a correct answer. What about sample 3? Do we see a shift? Yes. Who's the only factors in the tube? Pol2 and 2F. Would that be able to bind this TATA promoter without any other factors present? No, we already said we need 2D there and then all of the other factors in order. So it's also not more than one of the above. So this question required you to think about the sequential binding of these general transcription factors and then look at the data that we see up here. Here's a question for you. If sample 2 is TF2B, which one would be TF2D? Is TF2D a bigger complex than a single protein TBP? Yes, TBP is one of like 10 proteins that are part of TF2D. So if this lane is TBP, if just TBP is here and it shifts here, which sample would be 2D? Have to be 3, right? You would get a bigger shift. It's bigger. It migrates more slowly through the gel. Does that make sense? If I told you that 1 was TBP, 2D could be 2 or 3. We don't know. That makes sense to you? All right, so that's how we interpret a gel shift, and then just remember this nice little mnemonic. Question? Sample 1 could be TBP. It could be TF2D. It could be anything that alone can directly bind the complex. Do you have a question?
Yeah, so like if it's higher up with the probe, does it have more proteins now?
So only if you know proteins that are in the complex, because the size of the proteins could be different. So there could be more proteins, but they could be smaller. But what we do know is if we're comparing TBP versus TF2D, which has TBP in it, it's going to have to be bigger than TBP alone. So if it has one of the proteins that you know binds on its own, and then more proteins, a higher shift, it's going to have to be something that has more.
So in this one, like 1 would just be like TF2D.
It could be.
1, but then like 3 would be like TF2D. And
it could be, this could be 2D. This could be 2D plus 2A. Question?
I mean, following that logic, wouldn't it also be correct if we said that 2 is already 2F2D, then
Can Pol2 and 2F bind the DNA by themselves?
they need to be first, TF2F2D.
D needs to be there, A would need to be there, B would need to be there, and then 2, and then F could come in. Right? All right. So what we said is once we have identified a protein and the sequence that it binds to, we need to figure out, well, what is the function of that interaction? Is it actually regulating transcription? Is it activating, or is it repressing transcription? So one way to do this is in vitro, so meaning in a test tube, where we're asking, does the protein affect transcription from a template containing the element, the sequence that you identified it to bind to? So for this experiment, we would create a template that has a core promoter element and then your sequence of interest that the protein binds to, and then some kind of sequence that can be transcribed. Since we're looking at transcription, we also need in our test tube the RNA polymerase II complex, so all of those general factors in order for transcription to occur. We need ribonucleotide triphosphates for RNA to be made, and then the protein that you are testing. So you put all this together in a tube, and you're asking, when I have the protein versus when I don't have the protein, do I get a difference in the amount of RNA I make? So how am I going to detect the product? My product is RNA here. How could I detect it? How can I detect RNA? Right, and what experiment would I do here? I could use a probe, and what type of experiment to detect RNA? A northern blot, yeah. I could do a northern blot. I know the sequence in my template here that would be transcribed. I can design a probe. Could I also use radioactive nucleotide triphosphates and just directly look at the RNA that's made? Yes, you could also do that. So those are the two different ways you could detect it. Does this need to encode GFP or luciferase or anything? No, there's no ribosomes in my test tube. I'm not going to make the protein. This sequence can be anything as long as it's an RNA that you can detect. And here's an example of this type of experiment. So what the researchers did here is they have a template from adenovirus DNA versus SB40 DNA, so two different viruses, and they're testing the effect of a specific protein on expression from these two different promoters. So looking at the results here, which template is actually regulated by the protein? Yeah, SB40. Adenovirus, doesn't matter if I don't have the protein or I do have the protein. I get the same level of expression. Protein means nothing. Does that mean that protein doesn't bind to that promoter? Does this experiment tell me binding? No. What type of experiment would I need to do to test that? The footprinting experiment. Or you could do EMSA if you just want to know, does it bind or not? If I want to know the position it binds, I would do footprinting. And that was the second question right here. We can see with the SV40, it definitely makes a big difference when we add the protein for the level of expression we get. So that's an in vitro way to get a readout from the test tube of whether a sequence we put into that tube and a protein affected transcription. We could also ask that same question, but using an in vivo system, meaning inside a cell. Still asking the same question, does protein X affect expression of a reporter gene, but now we're just using a cell as the readout. So what you would need in this experiment is you would need a gene that encodes your protein. So if you're testing protein X, you would put like a strong, unregulated promoter in front of it just to make sure a lot of protein X is made in your cell. And then you would have your plasmid that has the reporter gene. And so this, now we do want to encode something that we can easily detect, like GFP. We would then have a core promoter sequence, and our element, our sequence that we've discovered is bound by protein X. We put both of these plasmids inside the cell, so this one so that we can make the protein, and this one so that we can test expression from our reporter. If the protein binds to that element and turns on transcription, then we would expect to get high levels of GFP, for example. If it binds and represses, we would get less expression than what we would get if we didn't put in the plasmid for encoding the protein. So in this kind of experiment, it's important to do controls to be able to compare. Do I see a difference in expression with the protein versus no protein? If it looks the same, plus or minus protein, then it's not regulating transcription. If expression is higher when I add the protein, then it is activating. If expression is lower when I add the protein, it is repressing. Does that make sense? All right, so those are sort of our functional assays. And then the final type of experiment that we'll start to talk about is a way that we can detect specific association of a protein with a DNA sequence in vivo, so in cells, in an organism, in a tissue. And this experiment is called a Chromatin Immunoprecipitation Assay, sometimes shortened as CHIP. Immunoprecipitation, what does that remind you of, or what does that mean? Immunoprecipitation. What are we going to use in this experiment? Antibodies, right? So antibodies, part of a normal immune response, but the beautiful thing about antibodies is that they very specifically detect a particular protein, sometimes even a differently modified version of a protein. They're very good at specifically detecting that. And so we'll see where the antibodies come into play in this type of experiment. So the goal of this CHIP experiment is to identify whether a specific DNA sequence you're interested in is associated with a protein or even a modified version of a protein in your cell type or your tissue of interest. What is happening in vivo? You're basically going to freeze the cell in whatever was going on in the cell under that particular condition. And the way we freeze it is we cross-link. So we use chemicals or sometimes UV to cause covalent bonds to form between proteins and closely associated DNA. So if a protein's bound to a piece of DNA, it's going to get cross-linked to it. The next step you do is you isolate the chromatin from the cross-linked sample. And then you're going to break that chromatin into pieces. So what do we mean by that? Well, if you've cross-linked your cell, you have your huge million base pair chromosomes all cross-linked to protein and all stuck together. And so you can use either enzymes or mechanical force, sonication, to break that chromatin into much smaller, manageable little chunks. Usually they'll end up being a few hundred base pairs long and still covalently bound to whatever proteins they were associated with. For example, histones. And the example shown here, they then wanted to isolate only the pieces of chromatin that were associated with the acetylated histone. So let's say histone 3 with an acetyl group on lysine 9. We actually have antibodies that are specific. Won't just immunoprecipitate histone 3, but only histone 3 when it is acetylated on lysine 9. So you use those specific antibodies to select the chromatin pieces that have your protein of interest. So these blue Ys, that's the antibody. The green indicates the nucleosomes that have that acetylated histone in it. And so the antibodies will bind to that protein epitope. And then what you do is you wash away everything else that was not bound by the antibody, and you have immunoprecipitated. You have pulled out that protein, and it's still covalently bound to whatever DNA it was originally associated with. The next step is to release or purify the DNA. You get rid of all the protein. And so now all you have is the DNA in the cell that had been associated with that protein of interest. All the other rest of the DNA, all the other genomic DNA that were not with histone 3 with that acetyl group got washed away. And now you can ask, the gene you're interested in, did it get immunoprecipitated? Was it associated with that acetylated histone? And the way you do that is simply PCR. You know the sequence of the gene you're interested in. If it was associated with that protein that got immunoprecipitated, there will be template for that gene. You will get a PCR product. If that gene was not associated with that protein, there will be no template. You will get a PCR product that is blank. Does that make sense? Those are the basic steps of a CHIP experiment. And we'll start class next week looking at what seems to be a very complicated example of this CHIP experiment, but we'll walk through it and make sure you know how to interpret these types of data.